Good afternoon to all. This is Nalini, working as School Assistant Biology in ZPSS Jallepalli, Mandal of Tirunaipalam, Khammam district. In the previous classes, you have learned about tissues, cells, and all. What is cell? It's a fundamental organizational unit of life is the cell. Cell is the fundamental organizational unit of the life is the cell. What is tissue? Can you define it? Tissue is a group of cells similar in structure and similar in performing functions is known as tissue. Tissue is a group of cells similar in structure and performing similar functions in. Now we are going to learn about dermal tissue in rheolith. Before going to know about dermal tissue in rheolith, we have to do one experiment. Before going to do experiments, we have to follow some steps. Those are A. Apparatus, materials required, precautions, procedure, and observation. These steps we have to follow to do experiment. What is this? A. Apparatus, material required, and precautions, procedure, observation. A. To observe the dermal tissue in rheolith. Apparatus, material required. Rheolith, microscope, watch glass, slides, cover slips, water, glycerin, brush, etc. Apparatus, material required are rheolith, microscope, watch glass, slides, cover slips, water, glycerin, brush, etc. Now, precautions. In the preparation of slide, we have to take precautions not to come air bubbles on the slide. To avoid these air bubbles, we have to put cover slips on the slide. Due to this cover slip, we not get air bubbles on the slide. Now, procedure. Take a fresh leaf of Rio or Billy plant Why we are taking Rio or beetle leaf? Because the stomatal structure is visible very neatly and perfectly in the leaves of Rio or beetle plant. So we are using these two plants. Procedure. Take a fresh Rio leaf, tear it in a single stroke, and we take a peel in the Rio leaf, and this Rio leaf peel put in watch glass with watered watch glass, and after that we have to take the peel with brush and the peel put on slide and put one drop of glycerin because to avoid water on peel. After we have to cover with cover slip without air bubbles. Cover slip used to cover the peel. Now we have to watch in the microscope this mount slide. We have to observe in the microscope. Observation. In the microscope, we have to observe single layered cells, which is dermal tissue and stomatal structure also. In stomatal structure, we observe this type of cells. In the microscope, we have observed this type of stomatal structure. Now, here we are observing these two guard cells, guard cells, chloroplast, stomatal aperture, adjacent cells, and inner wall. Inner wall. These parts we have observed in the microscope. Now we are going to do experiment. What is the aim of experiment? To observe dermal tissue in Rio leaf. Apparatus material required are Rio leaf, microscope, watch glass, slide, watch glass, water, cover slip and glycerin. Now take a fresh leaf of Rio and tear it in a single stroke. We observe white peel in the leaf. Now we have to take the peel, put it in water, and after take this leaf with needle and put it on a slide and put some drop of glycerin and cover it with cover slip to avoid air bubbles on the slide. we have to remove the excess of water using cotton or tissue paper. After that, we have to put under the microscope and observe it in the microscope. To view stomata clearly, I am using eyepiece lens 15x and objective lens 10x by 0.25 to view the stomata clearly. See children, we are observing slide closely. Watch. There is thick walled pore-like structures, that is stomata, which is located in the dermal tissue. You can also see stomata, dermal tissue clearly. By zooming now, we can see clearly the stomata appearing very clearly. We can observe dermal tissues also very clearly. Now we can adjust the light of the microscope to view stomata in 3D view. We adjusted the light. Now we can observe the stomata in 3D view. This is the structure of the stomata. It's appearing very clearly. There are 3 stomata visible to see. Now we can see this very clearly. By adjusting the objective lens as 40x by 0.65, in this view we can zoom very clearly. Here we can see stomata. It's appearing very clearly, and we can see only one stomata very clearly. Now look at this. Now I am adjusting the microscope resolution. We can see the pore. See, now you can see the pore. The guard cells and chloroplasts also. This is the stomata aperture. Next we can observe guard cells both sides of the stomatal aperture, and we can observe chloroplasts also. Number 2, chloroplasts. Number 3, inner walls. Number 4, stomatal aperture. Number 5, adjacent cells. This stomatal aperture can use to gaseous exchange and water exchange in the dermal tissue. Dermal tissue functions to protect the plant from injury and water loss. The dermal tissue covers the plant body except in woody shrubs and trees which have bark. Example for bark: tamarind, mango, neem, etc. The most common cell type in the dermal tissue is the epidermal cell. Generally, a thin waxy layer called a cuticle covers the epidermal cells and protects them. Example for waxy layered plants which have cuticle on their epidermal cells, those are xerophytes. In xerophytes we observe cuticle. Example: opuntia, aloe vera, etc. Other cells in the dermal tissue are guard cells. that surround the stomata, which are openings in the leaf. Gases and water enter and leave the dermal tissue through stomata. Stomata is the opening of dermal tissue. In the examination point of view, this topic is very important. In the examination, may ask this type of questions. Number one is explain the functions of dermal tissue. Number one is explain the functions of dermal tissue. First question. Number two, draw a. My special thanks to Commandant Suryapet DO Sir Shri Madan Mohan Garu to giving me this opportunity, and my school HM Sir Shri Maksud Ali Sir and my colleagues. I am very thankful to them. Thank you.